So you all went down to collect the algae. Will and Shayna were with the team going down there. How did you all collect it? Did you all have to wear any special protective clothing?
Yes, we did have to wear protective gear. The hazmat team was very nice to us. They gave us, okay, you can go out on the beach, you can collect, but you have to wear these booties and these gloves across your hands, you know. So we did that.
Did they require you to put it in special containers, or you had brought your own collection?
We brought our own collection materials because we had to preserve it in 3 different ways so we can analyze it back in the lab.
Right. And you mentioned that you collected both deep water algae and algae along the shore.
Well, the deep water, they were cast on shore. See, they were originally from deep water, and then with the currents and whatever, they come in the drift. So those were coated. So that means that wherever they came from, there was not a lot of oil.
Right. And what's the difference between the deep water algae and the coastal algae? Is there a physical difference, Shayna? Like if you're just looking on the shore, how would you know whether it was deep water algae or?
I think basically like deep water algae, you really can, that's, the one that's really at the bottom, whereas something on is, probably just floating on top, and you could probably see that it's probably been there for a while. And just by looking at the physical features, you probably can tell the difference.
Right. So how much did you all collect? I mean, 10 pounds, a ton? What?
We had a whole cooler full, several bags, so we tried to separate them a little bit.
Right. So once you get it back to the lab, some are heavily coated, the ones that came in from the deep waters, some are closer to shore. What do you all do? Just help us lay people understand when you bring these into your laboratory, what are you looking for and how do you find it?
Well, what we do, we make sections, we compare them, we look, or some of the cells, have they degraded or not? Do they look healthy? What is their vitality and things like that, their reproductive status. So probably it's gone, every time we would go, we probably would see something degenerate, you see? So we can compare to the algae that we had collected before the oil spill and see what grows there now, what is going to grow there next month. So we have very nice baseline data. And that's our strength, I think. And that's why we also just submitted one of those rapid proposals to NSF, Dr. Felder and I, because we have all that baseline data that we had gathered for years before the oil spill. And we always kept in mind that we needed that in case an oil spill pops up. And of course, unfortunately, it just happened.
Right. So often when there's a disaster, people begin to want to study that area, but without a baseline
you need to have something to compare it with. And that's our strength, because we were in charge of the algae, the algae group with seaweeds, and then Daryl Felder was in charge of the animals. He works on crustaceans, but we collected a whole bunch of other animals that he then would give to other scientists. So we basically have all the invertebrates covered, and we have all the seaweeds covered. So we have a very wonderful database. And
clear understanding of what the Gulf was like before this happened.
And the Gulf in the northwest. Then we have the northeastern Gulf. We collected in the southwestern Gulf and the southeastern Gulf. So we have it basically covered. Different times of the year we went. So.
Now, you mentioned that you and Daryl Felder just recently got a very quick NSF grant to be studying our...
Well, we hope... Yes, we submitted one. We haven't received... we don't know yet if it's going to be granted.
What would this allow you to do most immediately? To
recollect right away. In same areas we've been collecting before. And then... so this is a rapid grant. It's for quick response research. And that would then give us time just to see what's going on, to then write more long-term grants, you see? But just to see now what happens at different intervals of the year. So hopefully we will be able to see what kind of algae or animals recover from the Gulf, which one don't, because everything is going to change with all those bacteria that normally a lot of them... grow into those seeps, and we have those methane seeps, so they are there, but in limited numbers. You see, it's kind of restricted to some of those methane seeps. But now they're probably all over the place. So, like I say, they are, they're feasting on it. And how is that going to affect the whole dynamics of the Gulf, of all the interactions? Because the whole sediments have changed, the life forms have changed. Who knows? A whole bunch of algae may or animals may completely die out, and others may arrive and be brought in and flourish. We don't know it. We don't know yet, so that's why we have to study it.
So much to discover.
Yeah.
And students like Will and Shayna, you're here at a wonderful time with your skills, knowing how to work with the seaweed and the algae, and to be able to help people understand just where this is going. So, Shayna, do you like lab work?
Yes, ma'am. I really like it. Mm-hmm. It's really interesting. It's something different every day, and I'm really learning a lot actually working in Suzanne's lab.
Well, so, for example, you bring this seaweed, this algae in from Port Fourchon. You all bring it into the lab. What's your job? What do you do with it?
Basically, it's to preserve it in formula and to make sure that it stays preserved and everything stays okay with it.
And is it alive? It's alive when you bring it into
No,
the...
ma'am.
No, okay.
Yeah. But some of it is alive. No, when she does a lot of molecular. That's
and extractions.
Yeah, extraction.
And amplifying the DNA of it.
And what do you use to extract that from the algae?
Different type of buffers and DNTPs, just making a whole cocktail of it and stuff like that.
All right. Well, we can hear the delight in your voice. But that kind of lab
very
work.
good at it. Very good.
Well, to be an undergraduate and be involved in this, that says a lot about your skill. Now, Will, what was your dissertation on?
My dissertation was on the rhodomonates, which is a division of red algae, a group I should say, from the red algae, and it was on from the Gulf of Mexico. So I grew up in my doctorate on specimens from the Gulf of Mexico, so I have a real connection with it, I guess you would say.
Well, now you mentioned that you just went out and collected whatever algae you could find. Was there some red algae among the algae that you collected in Port Fourchon?
For the onshore, yes. We collected Bangia, which is a very simple red algae, and it can handle a lot more stress than the more complex red algae. And then inshore, like in the mangrove areas, we collected another type of algae like Bostrichia and Caloglossa, which we even described from that area.
All right, so lots of different types of algae, and this will give you a beginning sense of the effect, because that's interesting that you mentioned that some can take more stress than others, and that'll make a huge difference.
Exactly, because we don't know much how each group is going to react to the oil spill. Some may not be much affected by them, others will. So we need to find out.
Right. And also you mentioned that you're interested to find out how it will actually change these groups themselves, because you study evolutionary biology.
Yes, absolutely. Yeah.
And any sense of how it might change some of these species?
Well, there may be maybe there are some spores there, and that they have been kept at a spore level. But somehow they suddenly might disrupt and proliferate. We don't know, and take over. So it's really like in a bad B movie, something.
I was thinking
exactly.
That
like everything we can
aliens.
Everything that we can imagine probably is not imaginative enough for what's going on in real life. We don't know, because we don't know what happened there at a mile below the surface. And what is so terrible, because it's so deep. So what is horrible is when you look at the coastline, you see the stranded pelicans, the fish, the porpoises, the sea turtles, and it's heartbreaking. But this is basically the tip of the iceberg. It's probably because they can be helped. The beach can be cleaned. The marshes are going to probably recover, except, of course, they're going to die out now. And with the hurricane season, you're not going to have that buffer. But still, I think this is still minimal compared to what's happening in the deep areas. Because, like I said, all the base of the food chain is right there.